Now, sometimes diffusion by itself isn't enough to get things across the membrane. Sometimes diffusion needs a little help. Not active help, not work, because then that would be the active transport we're going to talk about in the next lecture. No, what we see is instead of allowing whatever to diffuse through the membrane, maybe it can't diffuse through the membrane, or maybe it'll just go too slowly through the membrane. It's timing. It just can't wait. The cells can't wait that long. So what we're going to see is sometimes there's going to be a little protein there to help. Having a protein help things diffuse. Still going from high to low. Okay, it's still diffusion. But we refer to this as facilitated diffusion or facilitated transport. It's diffusion, it's just now there's a protein helping it to occur faster. We see two types of proteins involved in this facilitated transport, this facilitated diffusion. First one being channel proteins. Channel proteins are exactly what they sound like, a channel. A channel, is a waterway where water can move through. Well, here a channel is the same thing. It's nothing more than a protein tube where things can move through it. So we're going to see, that channel, though, sometimes can be open, it can be closed. If it's open, only certain things can get through it. Carrier proteins. Think of these as turnstiles. Only certain things can go through the turnstile. Other things won't fit, or they have the wrong shape. When you look at channel proteins are composed of hydrophilic amino acids, so they're going to insert, they're an integral protein. But instead of being a clump, like I said, they form a tube that passes all the way through so that things can rush in, things can rush out. Some of the times these channels are always open. Other times they may be able to open and close depending upon certain cues or certain needs of the cell. We call these gated channels. They open, stuff can pass through, in and out of the cell. They close, nothing gets through. And that's basically what they do. Some great examples of these are what are known as aquaporins or aqua channels, water channels. Water channels allow water to move in and out of the cell very quickly. Yes, water can pass through the membrane, but, there's ways to make it happen faster. Ways to allow it to happen faster is what I should say. We also see that in muscle cells, there are what are referred to as gated ion channels. Under normal conditions, these channels are closed. Something will bind to the channel, will open it up, and then ions will rush out. Specifically with muscles, we see that calcium will be stored in the sarcoplasmic reticulum. Signal comes down through the nerves to the muscle fibers, which will, through a series of steps, cause the gates to open on these channels. Calcium will rush out and flood the cytoplasm. This will then trigger, through a few steps, a muscle to contract. The carrier proteins, well, they're a little bit different. Okay? They don't allow things to rush in to rush out. They're generally going to be one direction. And they're going to be highly specific. Remember, these are the turnstiles. So only things with the correct shape can fit through the turnstile. Remember, shape dictates function. Molecules or whatever will have the correct shape, thus they can fit in the turnstile. And what these do is just this. Open to the outside, open to the inside, outside, inside, outside, inside. Whatever can fit down into the carrier protein, whatever has the correct shape, will cause the carrier to do this, releasing it inside the cell. Once it's released, it snaps back. There you go. Like I said, that's why I say a turnstile. Now we're going to see sometimes the turnstiles is going to need more than just one thing to fit into it to cause it to change shape and then snap back. But it's still going to be highly selective. I'm not going to spend a lot of time discussing osmosis. I'll let you read that in the chapter in osmoregulation.